and that's where sperm are made in seminiferous tubules. And really these, I'm going to blow it up to a higher level, but each one of these is an individual seminiferous tubule. And then in between seminiferous tubules, you have, let me see, well here we go, raise it a little bit. Not the best slide for these, but you see like how in some of these you have very distinct cells in between the seminiferous tubules. They go by a couple different names: Leydig cells, interstitial cells, or interstitial cells of Leydig. But these are all the cells that secrete testosterone. So this is the primary, testes, primary source of testosterone. And let's blow this up to one level here, get a closer look. And this is mostly just memorizing the names of the cells and location of where the cell is. So every seminiferous tubule is a series of developing sperm cells, okay? And the most primitive cells are on the outside. These are actually sperm that have just been released into the lumen of a seminiferous tubule. Then the seminiferous tubules are going to lead to a bunch of other named tubules. Eventually those tubules will lead to the epididymis, which will eventually lead to the vas deferens, etc. All these different tubes eventually lead to the urethra and out of the body. Okay? So you have a basement membrane, it's a little acellular anchoring layer, and then on the very, very outermost cells are going to be the most primitive, less differentiated, either spermatogonia or primary spermatocytes, which still have diploid number of chromosomes, or 2N. By the time you get into the middle here, under the influence of puberty hormones, FSH, LH, and in combination you need testosterone for spermatogenesis. So those anterior pituitary hormones that we were talking about, FSH and LH, and testosterone, all start stimulating meiosis to begin. And meiosis, the first place where meiosis 1 occurs is between a primary spermatocyte and a secondary spermatocyte. So a secondary spermatocyte is the first time meiosis 1 has been completed, and now you have the haploid number of chromosomes. So basically, on the periphery, spermatogonia and primary spermatocytes, by the time you're somewhere in the middle here, you're going to start seeing secondary spermatocytes. And haploid, 23 chromosomes. By the time you're at the kind of like the periphery of the lumen, now they've differentiated into spermatids. Spermatids are still N or haploid, 23 chromosomes. So literally the cells right here are spermatids. And they're not quite sperm yet, they're still in the process of developing. There are these cells in there we'll be talking about called Sertoli cells that actually carry these developing sperm towards the center or the lumen of the tube, and then release the spermatid into the center. And really the difference between a spermatid and a sperm is a sperm has grown a tail, and a spermatid does not have a tail yet. So these are actually early stage sperm that have been released into the lumen of the tube. They're not capable of reproduction yet. They have to mature for like up to around 2 weeks in the epididymis. They have to learn to swim, essentially. So again, each one of these is seminiferous tubule. You got a basement membrane that you can't really miss. Out here, you'd have either spermatogonia or primary spermatocytes. In the middle here, you're looking at secondary spermatocytes. They've now just finished meiosis 1. They have the haploid number of chromosomes. And then eventually in the periphery, you're calling them spermatids. And at some point, they get released into the lumen. Now they're sperm. They just have to wait a while and grow up and mature. That's pretty much it. Again, you got your Leydig cells or interstitial cells that secrete testosterone. If you look at like any of the textbook or lab book drawings of the testes and seminiferous tubules, they look exactly like the slides. So very seldom do you see something that looks just like it looks like in the textbooks and various pictures. But that's all there is to that. We'll go into more details on meiosis in the lecture.